Hello, my name is Paul Roberts, and I'm going to be talking about the work we're doing at the Monterey Bay Aquarium Research Institute, developing a high-resolution camera array for deployment on autonomous vehicles. And this work is funded by the Moore Foundation in collaboration with the Central Northern Coastal Ocean Observing System. So I want to motivate this by thinking about how we monitor a changing ecosystem. And I'm showing here an illustration from an experiment conducted recently at MBARI, where we were studying the vertical migration of plankton in the bay. And the point is to show that in order to get a synoptic view of an ecosystem process, we need a lot of different platforms to do that. Here we have ships, autonomous vehicles, ROVs, bottom-mounted acoustic systems, moorings. But out of these, the systems that can go out for longer periods of time and do that at a low cost create an opportunity where we can imagine adding imaging systems to them and getting a much larger coverage, both in time and space, of an ecosystem. So thinking of these autonomous platforms, it creates both a challenge and an opportunity to deploy imaging systems. The integration is quite a bit more difficult and involved, but it opens up new possibilities, and that's what we're targeting here. So what are the benefits of imaging plankton on an autonomous platform? Well, this is an example here of an image of a few liters of seawater from the Monterey Bay. And from this image alone, we could start to get information about the who and what is there, how many there are. And if we add scale to the image, then we could say something about the projected size of the objects in the image. And if we add three-dimensional information, which I'm showing here by two different views of the same scene, then we could start to say what are the distances between objects in 3 dimensions, so proximity, what is the actual size and orientation of the objects, and then lastly, we could turn these into statistics about the 3-dimensional environment that most of these plankton and particles live in. Now, in the last few decades, there's been tremendous work on plankton imaging systems. This is an example from Lombard et al., who did a nice review of the available tools. And this is shown as the distribution of different imaging systems as a function of their size range. So on the horizontal axis is the object size that can be imaged, and the dashed lines are the nominal size range that the imaging system can work over, and the red lines are the quantitative range. And these systems employ a lot of different imaging methodologies, flow cells, free space, telecentric, light sheet, holographic, and they've really progressed to the point where there are lots of good options. And when we look at the subset of these, which can work with water samples collected in situ, it's a little bit smaller. There's still a number of them. But if we go a bit further and ask which ones can provide 3D spatial relationships about the water sample, so where were the objects relative to each other in 3 dimensions, then it's really just the holographic systems that also collect 3-dimensional data. So there's some motivation to look at new technologies that can be developed to go on autonomous vehicles and enable 3D imaging in situ. So what we're interested in here is a system called AyeRIS. It stands for the Autonomous AyeRIS Remote Imaging System. It's kind of a mouthful. This stems from our work in developing light field cameras to go on cabled instrument platforms. And if you're interested in learning more about the cabled instrument, Joost is giving a talk in PIO2, studying flow inside and around deep sea animals in 2D and 3D using novel imaging techniques. Here we're looking at how do we adapt that technology to work on autonomous vehicles. And we're envisioning this as a camera array and strobes that would be integrated into the payload of a AUV, imaging a volume of water that's out in front of the vehicle of several liters with 50 micrometer resolution, and allowing us to get quantitative imagery in real-time processing. So for the AUV, we chose to target the MBARI's long-range AUV. This is a very mature system, has over 22,000 hours of offshore operation across 12 different vehicles. It's long range and endurance on the orders of weeks. and it's designed specifically to support collaborative operations with vehicles communicating with each other during a mission. And it also has the advantage of being able to turn off the propeller and drift with the flow. So it's a great platform for plankton imaging. We're using a camera array. The camera array has a lot of advantages. It's flexible. It enables very standard multi-view processing, which has been developed quite extensively in other areas. The cameras and lenses can be changed easily, so we can support imaging at different scales and also different spectral ranges depending on how we configure the instrument. And then finally, it leverages some of the advances in drones and self-driving cars that have come about and enabled the compute technology to keep up with the increasing number of pixels produced by camera arrays. So this is a schematic here of the payload of the long-range AUV with the AyeRIS imaging system integrated into it. We have a flat acrylic viewport for the camera array. Surrounding the camera system, we have an array of strobes. This is a photo of one on the bottom right here. They're very high lumen output, about 30,000 each, and there's 10 of them. And these are a custom strobe that we designed and really had to design to be able to fit it inside of the 12-inch diameter of the vehicle payload bay. And we have the array of high-resolution lenses. These are Fujinon 23 megapixel. 6 of the lenses that form the hexagon are 16 millimeter and one is an 8 millimeter. So the central lens is imaging a larger field of view than the other 6. Behind that, we have the imaging sensors themselves. These are made up of Sony IMX183 20 megapixel imagers. So across the whole array, we have 140 megapixels. And then recording all those pixels and processing them are 2 Jetson TX2 compute modules. This is a photo of the carrier board, which has a 4 terabyte solid state drive added to it. Then below that, we have a custom controller and strobe ballast. And this provides energy for the strobes, timing synchronization between the cameras and the strobes, and then monitoring of power draw and environmental sensors inside the housing. And then lastly, we have the 2 48-hour lithium-ion battery packs. And these ended up being important in order to buffer the vehicle power system from the very high instantaneous power that the strobes are drawing. So after assembly and some testing, then we go into the test tank and do calibration with a standard checkerboard target, and then confirm vehicle operations with the imaging system firing strobes and then ballasting. Once that's ready, then we can go into the field. So last year we deployed the AyeRIS imaging system for over 60 hours in Monterey Bay, and we tested a number of different vehicle behaviors to see how it would impact image quality. We did circular profiles, yo-yo transects, station keeping, and drifting with the flow. And we also tested different processing modes, so storing all the pixel data locally, up to 7 terabytes, real-time particle detection and publishing to shore, so we could see some statistics of particle distributions. And then lastly, a really fun concept of supervised autonomy, where we detect objects while the AUV is subsurface, and then send a low-resolution version of the image up to a surface vehicle, which then sends it to shore. And this enables then an operator to make a decision to change the mission based on what they see in the imagery. And this is enabled using tracking between the surface vehicle and the AUV. And if you're interested in that work, Yvonne is giving a talk, OT05, a reinforcement learning approach for underwater target tracking optimization, where he'll talk more about how that tracking enables the 2 vehicles to stay close to each other. So this is an example of some of the multi-view data, just taking single frames from 3 of the cameras in the array and looking at one of the detected objects. On the top, I have the location of where the images came from in the array. And on the bottom, is a zoomed in view of the object. So this is a larvation that's about 1.5 centimeters. We're getting good detail of the house and also the animal. We can see good correspondence between features on the animal and then also the particles in the background. So the next step is to think about how we detect objects in real time and localize them in 3 dimensions. So what I'm showing here is a composite image of 6 of the cameras that form the hexagon in the array. Each camera is represented by a different color in this image. And the first thing we do is detect an object in one of the camera images. And from that, we get a bounding box, timestamp, camera ID, and then properties about the object like morphological features. From there, we have to solve the correspondence problem. And this is simplified in this case because of the multi-view geometry. So since the cameras are arranged on a hexagon, we get this hexagonal pattern. And you can see that in the image here, where the same object is viewed from different cameras. It shows up as a repeated hexagon with different colors. And I've drawn some triangles over the corresponding points. So we search along the triangles, and that simplifies the correspondence matching and also makes it more robust. Once we have that, we can add the 3-dimensional location to the detected bounding box. And then from there, we can go and distribute these data to different places. So we might send it to the vehicle for real-time decision-making, log particle statistics to distribute when the vehicle's back at the surface, or record ROIs to show when the vehicle's recovered or when it's back at the surface by transmitting over a cell network. So these are some examples here of the regions of interest that are detected. The top row is from 0 to 5 meters during a nighttime yo-yo transect. We can see fish, pyrosomes, salps, copepods, other crustacean. Going a bit deeper, between 50 and 70 meters, we can see it's pretty much dominated by crustaceans. And going even deeper, down to 110 to 120 meters, we see different types of larger gelatinous plankton like siphonophores and larvaceans, different species of fish, some shrimp. And this is a 10-centimeter scale bar here for reference of all these images. But the point is to show that the system is actually capturing a pretty wide range of types of animals, which is great. So we're able to see a lot of the larger species in different taxonomic groups with enough detail to do classification to a functional group level. We also looked at particle detection and particle size statistics. So this is just an example. There's a lot of work to do in this area, but this is showing some of the capabilities. So we, on the top row, have 3 different images from different depths at 5 meters, 50 meters, and 110 meters. And on the bottom are example histograms of particle size in terms of equivalent spherical diameter on the horizontal axis and the number detected in an image on the vertical axis. And these are averaged over 30 images. And so we can see changes in the particle size spectrum as we go from the surface down to 110 meters. We see about 10 times as many particles at 110 meters compared to 5 meters. And the shape of the size spectra is changing a little bit too. It's getting rounder instead of the more typical log linear relationship. So there's a lot more work to do on this. We're just getting started. One of the areas of future work is to develop more robust object detection models. And for this, we're going to use data in FathomNet. Kakani Katija is going to be giving a talk about that in IN05, an open source image database to train artificial intelligence algorithms that help us understand our ocean and its inhabitants. We're also interested in making the vehicle and the imaging system able to target biological features, so have it detect elevated abundance of certain taxa and then sample that depth or that part of the water more densely. and Erik Orenstein's giving a talk about that. OD13, autonomous detection and sampling of biological features with the AyeRIS imaging system on an AUV. We're interested in developing more robust 3D particle statistics that can be sent to shore when the vehicle comes to the surface, so we can learn about the particle distributions subsurface as a mission is unfolding. And then lastly, we're developing deeper water versions to go all the way down to 6,000 meters. Thank you very much.